Hello everyone, I am SARITA RAWAT, Assistant Professor, School of Pharmacy, Suresh, Gyan, Vihar University, Jaipur, Rajasthan. Today we will start our lecture on pharmaceutical microbiology, and today's topic is Introduction to Scope of Microbiology in Pharmaceutical Industry. Now, learning objective of today's lecture is to study about the introduction of microbiology and scope of microbiology, development of microbiology, scope of microbiology in pharmaceutical industry, retention with other allied sciences, and its relation with the other allied sciences. Now, learning outcomes of today's lecture is you are able to know about the basic introduction of microbiology, development of microbiology, scope of microbiology, and relation with the allied sciences of microbiology. Now, content included introduction part, history, branches of microbiology, and scope and importance of microbiology. Now, when we talk about the basic introduction of microbiology, so microbiology is the study of microorganism that is usually less than 1 millimeter in diameter, which requires some form of magnifications with the help of microscope, so to be seen clearly. So we can say microbiology is the study of that microorganism which are not like visible with the naked eyes. We need microscope to see them. For example, virus, bacteria, fungi, algae, and protozoas. Now, the word microbiology was derived from Greek word that is micros, which means very small, and bios, which means life. So, it means the small life means small organism life, to study about the life of small organism. So, it is the branch of science that is connected primarily with the biology of microorganisms and their effect on the living organism. For example, bacteria, protozoa, algae, fungi, and viruses. So, microbiology may be pure and applied. So, pure microbiology is included bacteriology, mycology, protozoology, physiology, parasitology, virology, nematology, cellular microbiology, evolutionary microbiology, and molecular microbiology. It's also include the various other studies. Now, when we talk about the applied microbiology, okay, so it includes the medical microbiology, pharmaceutical microbiology, industrial microbiology, food microbiology, and social microbiology, and water microbiology, air microbiology, and microbial biotechnology. So these are the area where we follow the applied microbiology, and our subject is pharmaceutical microbiology came under the applied microbiology. So when microbiology concept, process and techniques are applied to the pharmaceutical operations, that subject is called pharmaceutical microbiology. Now, what is pharmaceutical microbiology? So, when we talk about the pharmaceutical microbiology, it is the combined field of microbiology and pharmacy, wherein study of microorganisms that are related to the production of antibiotic, enzyme, vitamins, vaccines, and other pharmaceutical products are detailed. So it also deals with the causative agent of the disease, diagnostic process for the identification of causative agent and preventive measures of any kind of drug. So as you can see, the pharmaceutical microbiology include the production of antibiotic, enzyme, vitamins, and all. It also deal with the causative agent. Which is responsible for that particular disease, and also the identification of the causative agent of that particular disease. Now, when we see the significance of microbiology in pharmaceutical sciences, so our cell have been fighting microbes that enter our body and commonly exhibited by the pus formation and inflammation of the wound. So, microphages play role in the immune system because they are capable of ingesting the microbes that enter our body through the open wound. So, however, some microbes adapt and mutate rapidly, resulting in the infectious disease like HIV. So, the antibiotic, vaccines, etc., produced to get resistance from that microbes. So, on the contrary, microbes help us like lactobacillus with function of our digestive system. So, we basically the main significance of pharmaceutical microbiology is to make a protective layer of various to protect the human from the various kind of disease. Now, the factor involves in pharmaceutical microbiology are pharmaceutical microorganism, pharmaceutical contamination, pharmaceutical product and microbial risk, microbial limit test and assay, microbial spoilage and prevention, antimicrobial prevention, disinfectant and sterilization techniques, and clean room and control techniques, drug quality control, validation of the test method, drug safety and pharmacovigilance, sterilized pharmaceutical manufacturing, and biological indicator. So these are the main factor that involved in microbiology, pharmaceutical microbiology, and we have to cover all these topics to understand better the pharmaceutical microbiology. Now, microbiology may be interested in the specific type of microorganism, like virology is the study of virus, bacteriology is the study of bacteria, physiology is the phycology is the study of algae, mycology is the study of fungi, and protology is the study of protozoa. Now, microbiologists may be interested in various characteristics or activities of microorganisms such as microbial morphology, microbial cytology, microbial physiology, microbial ecology, microbial genetics and molecular biology, and microbial taxonomy. So these are the various fields where we can broadly study about any kind of microbiology, and we will get more knowledge about that particular microorganism. Now, objective of pharmaceutical microbiology is to ensure safety and efficacy of pharmaceutical product. It embraces of validations of disinfectant, evaluation of the efficacy of disinfectant in suspension or surfaces and throughout the field trials. Now, it offers protocols and techniques associated with the operations and assurance of clean room, aseptic room, and controlled environment for preventing any possible microbial contamination, and introduces the risk assessment and practical contamination control strategies. Now, Louis Pasteur is regarded as the father of microbiology, whereas Robert Koch is the father of medicinal microbiology. So, Coming to some historical perspective, so Antony Leeuwenhoek is the first to observe living microorganism, and his single lens magnified 50 into 300x magnifications. So as you can see, this is the picture of the first microscope that is seen by the Antony Leeuwenhoek. He manufactured it, then he see the single lens magnified 50 into 300x magnification. He see the first living microbes in this particular microscope. So he was the first person to actually see the living microorganisms. So between 1674 to 1723, he wrote a series of papers describing his observation of bacteria, algae, protozoa, and fungi. So as you can see, this particular first microscope contained the lens and then sample holder, which also nowadays also it is there. Then focus knob, and then this is a simple transducer. So as you can see, this is very simple microscope and first ever invented microscope by the Antony Leeuwenhoek. Now, coming to the Robert Hooke, Robert Hooke, one of the most important discoveries of biology occurred in 1665. So, with the help of a crude microscope, when Robert Hooke started that smallest structure units are cell. So, he discovered that the basic structure unit of life is cell, and the smallest unit of life is cell. Now, coming to the Louis Pasteur in 1822 to 1995, he was a French organic chemist, and he considered as the father of modern microbiology. And he processed that fermentation process. He discovered the fermentation process and gave a theory of beer and wine not produced without microbes, and microbes cause spoilage, and demonstrated the gun cotton experiment. And he also differentiated places and different concentration of microbes, and he developed the pasteurization process by boiling of milk in 50 to 60 degrees Celsius, and he also developed the rabies and anthrax vaccines. Now, coming to the Robert Koch in 1843 to 1910, he was a German physician and first to demonstrate the role of bacteria in causing disease. And the first isolated bacteria he discovered Bacillus anthracis. Okay, so discovered Mycobacterium tuberculosis in 1882 and Vibrio cholera in 1883, and he was also awarded with the Nobel Prize in 1905, and he also developed the relationship between the microorganism and the disease. So he invented that the disease are caused by some particular microorganisms, like tuberculosis is caused by Mycobacterium tuberculosis, and cholera is caused by Vibrio cholera. So, Coates postulates that consist of the following 4 rules. He followed the 4 rules that are: the microorganism must be identified in all individuals affected by the disease, but not in the healthy individuals. So, whenever any person suffers from some serious disease, so it is very important to identify the particular causative agent, particular microorganism that is responsible for that particular disease. but it is not followed with the healthy individuals. The microorganism can be isolated from the diseased individual and grown in the culture. So, if any person is suffering with the particular disease with that causative agent is particular microorganism, so we can isolate that bacteria from that person, and then we can grow it in a culture. Then, when introduced into a healthy individual, the culture microorganism should cause the disease, and then when we transfer that particular microorganism in a healthy individual, so it can cause the disease to that healthy individual. So the microorganism must then be re-isolated from the experimental host and found to be identical to the original organism. Then again, when we isolate that particular microorganism from that particular individual which we had ingested the particular microorganism, and then we isolate it, and when we identify that, so we will found that that is the same microorganism which has diseased the previous human. Now, what is the role of microorganisms in disease that are discovered by various scientists in various years? So, Oliver Holmes in 1773 to 1843, he showed that species could be transmitted by hand of medical student and many cause diseases. And then M.J. Barclay in 1845, he demonstrated that the great potato blight of the Iceland was caused by a fungus. Then Louis Pasteur in 1822 to 1892, he showed that the peregrine disease of silkworm was caused by a protozoan parasite. Now, what is the scope of microbiology and what are the importance of microbiology in uh, like in the pharmaceutical industry or in the point of view of pharmacy? So the first is production of antibiotics. So it is very important role of microbiology nowadays. So two-thirds of antibiotic are produced from microorganisms. Pharmaceutical or industrial microbiology concerned with the isolation of antibiotic, producing microorganisms from natural environment like soil, water, and many antibiotics are isolated from natural microorganisms by the process of fermentation. For example, penicillin from the Penicillium species. Then the second use or scope of microbiology is production of enzyme, vaccines, biosurfactant, pharmaceutical product. So microbial cell produce intracellular and extracellular enzyme. Those are important for the pharmaceutical fermentation. For example, amylase and protease. Then biosurfactant have lots of application in the food industry, agricultural industry, industrial cleaning, leather, paper, textile industry, and also in the pharmaceutical industry. So many microbial cells convert raw material or substrate into the valuable organic compound such as butanol, ethanol, and etc. So the third use is diagnosis of disease and treatment. So it is very difficult. Tests are used to detect infectious microorganisms. For example, ELISA test is used, and Vidal test is also used to identify the causative agent in the particular disease. Antimicrobial susceptibility test is mainly used for the selection of antibiotic for the treatment of particular microbial infection in a person. Then the fourth use is treatment of industrial waste material. So some microbial species are used for decomposition of industrial waste material and organic compound, for example actinomycete and fungi. So anaerobic bacteria decompose of organic matter by yielding some organic molecule such as acid alcohol, glycerol and amines. Now, the another use is plant growth promptations. So, many microbial cell plant in soil play the important role in the soil fertility, herbicidal resistance, change in the protein and oil content, and enhance the quality of the plant in, and product, for example, rhizobium species, zotobacter species. Then, many nitrogen fixing soil microorganisms are used for providing nitrogen from the growth of the plant, germination of the seed, and flavoring the fruiting of the medicinal plant. So, they are also used to grow the particular kind of plant. Then the another use is sterilized product preparation. So it deals with the preparation of sterile room, aseptic techniques, detections of microbes by sampling, and sterility testing of different sterile preparation. So, what is sterilization? So, the next use is sterilization physical method. As you can see, this is in this picture. This is the physical method of sterilization. So, you can see this is your hot air oven, and then this is by the sunlight, with the help of sunlight radiations and autoclave. So different pharmaceutical products are sterilized by killing of microorganisms. The process of killing of microorganisms is known as sterilization, and all the sterilization process may be validation by using biological indicator. So the next use is steroid biotransformation. So steroids are physiologically active compound of complex structure. For example, cholesterol, ergosterol, testosterone, progesterone, and etc. So important steroid can be produced by the microbial transformation of naturally occurring steroid. For example, Streptomyces species, Rhizopus species, Acephalus species, and Penicillium species. Then the next use is identification of microorganisms. So one of the vital function of pharmaceutical microbiology is identification of microorganisms and found in the product and the manufacturing environment. So the microorganisms are isolated and identified by the morphological, biochemical, cultural, and microscopic characteristics and genetic studies. So the next use is testing of pharmaceuticals, so raw material and finished product testing. So the presence of microorganisms like E. coli, Salmonella species, Pseudomonas aeruginosa, and Streptococcus aureus in the raw material and finished product may deteriorate the efficacy and potency of the product. So test the presence of microbes are described in the IP, USP, EP and BP. So limit on the total number of variable microorganisms in given product and involved in explosion of the specific pathogen it is used, and water act as a vehicle. Always test for the TMC, total microbial count. Now, also in the development of vaccines and antistria. So, Edward Jenner in 1796 discovered that cowpox vaccinia introduced protections against the human smallpox, that is called producer vaccination. Then what is vaccination? So inoculum of healthy individual with weakened or attenuated form of microorganisms that would otherwise cause disease to provide protection of active immunity from the disease upon the later exposure. Now, Pasteur and Rollux reported that the incubating culture longer than the normal in the lab resulted in attenuated bacteria that could not longer cause the disease. And working with the chicken cholera caused by Pastula multipoda, they noted that the animal injected with the attenuated culture were resistant to the disease. So another use of microbiology is microbiological assay of antibiotic. So the antibiotic bioassay provide a potentiality for the overall bacterial activity of an antibiotic preparation. And agar diffusion plate assay and turbidometric assay also used as a microbial assay. Now, evaluation of disinfectant. So, microbiology, pharmaceutical microbiology also used for the evaluation of disinfectant. So, effective cleaning and disinfections of manufacturing facilities are crucial to the achievement and maintenance of high quality standard required for the medicine and medical devices. So, phenol coefficient test, cup plate method, and turbidometric method are the calcitrics test to evaluate the disinfectant. Now, the another use is antimicrobial preservative efficacy testing. So, different type of pharmaceutical product are cosmetic are protected from the attack by the microorganism by using some particular preservative. For example, methyl, ethyl, propyl, butyl, parabens, and sorbic acid. So, the preventive should be non-toxic, easily available, and have broad spectrum of activity. Now, the cup plate method, turbidometric method, and total viable count techniques are also used to determination of antimicrobial efficacy of the preservatives. Now, endotoxin testing. So, microbiology is also used in pharmaceutical industry for endotoxin testing. So, bacterial endotoxin or pyrorane have a number of physiological effects following intravenous injections such as fever, activation of cytokinin system, endothelial cell damage, and intravascular conjugations. So, the bacterial endotoxin test, that is BAT test, is most important in microbiological laboratory, and indeed they involve the quality control of parenterals and medical devices. And last test, gel clot method and chromogenetic endpoint method, kinetic turbidometry assay and kinetic chromogenetic assay are also used to detect the endotoxins. Now, it is also used in the test to support of the sterility assurance system. So, sterility test mainly applicable in running an aseptic area and microbiology lab. So, it is mainly performed in the laminar airflow cabinets or parenteral preparation and other sterilized pharmaceutical to detect the presence of microorganisms, and bioburden testing is used to estimate the number of microorganisms in the product prior to the sterilization. Now, what are the merit of pharmaceutical microbiology? So, when we see its merit, it is highly reliability, it has product safety, faster time to market, more innovations, and it helps in type of microbic drugs and their applications areas are known, and the effect of antimicrobic and non-antimicrobic drug on the immune system are known, and with the help of this, GMP and GLP applications regarding microbiology in the pharmaceutical industry can be known. So these are some merits of pharmaceutical microbiology. Now, talking about the demerits of pharmaceutical microbiology, so it time required. It need to time required. Like their process is very long and time consuming. They have a limited accuracy. They have transfer errors and the data entry errors, difficult entries counts, and new drug are expensive also. Then adverse drug reactions and side effects can also occur. Then some medications have low therapeutic index. Now, when we talk about the pharmaceutical contamination, so pharmaceutical contamination involves the introduction of microbes into the pharmaceutical finished product. So, source include drug manufacturer, raw material use, environmental sources, cleaning equipments, packaging, containers that are frequently reused, and repackaging of the product and processing, storaging, and transportation. So these are the factors, these are the various steps where the pharmaceutical contamination can be occur. And then factor affecting microbial spoilage of the pharmaceutical product include type and size of the contaminated inoculums, factor related to the pH, moisture content, storage temperature, packaging, designing, and very other factor also. So as you can see, microbial contaminations sources are very vast, and we have to check out all these things, and we will study all these topics in our pharmaceutical microbiology and how to control these kind of contaminations. Now, coming to the quiz, we have concluded some questions regarding to this particular lecture, so you can answer these questions easily while going through the lecture. So, who is the first person to actually see the living microorganism? Second question is, who is considered as the father of modern microbiology? Then the third question, the first microscope was invented by. Then the fourth question, classroom microscope was invented by. A photograph which is taken from a microscope is called. Then the genetic material in viruses, and what is bed test, why we do microbial assay for antibiotic, how can we test pharmaceutical formulation, and what do you understand by evaluations of disinfectant. So these are the 10 questions which is related to our this lecture. So going through the quick revision of this lecture, you will get all these answers very easily. So now we have covered today about the basic introduction of the microbiology. So as you can know, what is microbiology? So microbiology is the study of microorganisms which usually less than 1 millimeter in diameter and which require some forms of magnifications with the help of microorganisms. Now it includes like virus, bacteria, fungi, algae, protozoas. Now we have covered that it will microbiology is made up of two words, microns, which means smalls, and biases means lines. And microbiology may be pure and applied. So pharmaceutical microbiology comes under the applied microscopy. So when the microbiology concept, process, and techniques are applied to the pharmaceutical operations, so the subject is known as pharmaceutical microbiology. Then what is pharmaceutical microbiology? So it is the combined field of microbiology and pharmacy, where the study of microorganisms that are related to the production of antibiotics, enzyme, vitamins, products are like detailed. So it also deals with the causative agent which cause the particular disease in the person. Now the significance of microbiology, and then with the factor involved in the pharmaceutical microbiology, so it include the pharmaceutical microorganism, then pharmaceutical contamination, pharmaceutical product, and microbiological microbial risk, then microbial limit test and assay, microbial spoilage and preventions, microbial preventions, and disinfectant and sterilization techniques, then classrooms and control techniques, then drug quality control, validation of the test method, drug safety and pharmacovigilance, sterilized pharmaceutical manufacturing, then biological indicators. Then microbiology may be interested in the specific type of the organism. So virological study of virus, bacteriologist study of bacteria, phycologist study of algae, and then mycologist study of fungi, and protologist study of protozoa. So then we have covered the various history perspective of microbiology. So as you can know, Antony Leeuwenhoek is the first who observed the living microbes, and he was the first person who actually see the living microscope microorganism. So then Louis Pasteur came, and he also developed the pasteurization process. Then Robert Koch came. Robert Koch is a German physician, and he was the first who demonstrated the role of bacteria in causing disease. And he first isolated the Bacillus anthrax. Then Olivier Holmes came in 1773 to 1843. Then M.J. Berkeley came in 1845. Then Louis Pasteur 1822 to 1995. Then scope and importance of microbiology include production of antibiotic, production of enzyme and vaccines, biosurfactant, pharmaceutical product, then diagnosis of disease and treatment of industrial waste material, then plant growth promotions, sterilized product preparation, then sterilization, then steroid biotransformation, identification of microorganisms, then testing of pharmaceutical, which include raw material and finished product, then development of vaccine and antresia. Then what is vaccinations? Then microbial assay of the antibiotic, evaluation of disinfectant, antimicrobial preservative, efficacy testing, then endotoxin testing, then test for the support of the sterility assurance system. Then we have covered the merits of pharmaceutical microbiology. So it includes high relatability, product safety, faster time to market, and more innovations, type of antimicrobic drug and their applications areas are known. Then the effect of antimicrobic and non-antimicrobic drug on the immune system are known. Then GMP and GLP applications regarding microbiology in the pharmaceutical industry can be known. So demerits include it required lots of time, it's required limited accuracy, transportation transfer error also there, then data entry errors also there, then difficulty in interism codes also there, then new drug and a new drug invention is very expensive because it take very like a long process, then adverse drug reactions and side effects are also there. So some medications have low therapeutic index also. Then we have seen that pharmaceutical contamination. So how can be the contamination can be occur in the pharmaceutical? So it can be by the environmental source, then cleaning equipments, packaging containers at the frequently reused and repackaging of the product, processing and storaging, transportation of the product. So there can be contamination occur. Now, I think you got your all answer of all the 10 questions. When we talk about the particular answer we have, so like when we talk about who is the first who is the first person who actually see the living microscope, microorganism in microscope? So our answer is Antony van Leeuwenhoek. Then when we talk about who is considered as father of modern microbiology, so that is Louis Pasteur in like 1822 to 1895. Then the third answer is like a person first microscope was invented by, so that is our Robert Hooke. Then the classroom microscope was first invented by so F. Jensen and Z. Jensen. Then the another question is like a photograph was taken from a microscope is called micrograph. Then the genetic material in a virus is DNA and RNA. So these are very simple questions and answers. Now, when we talk about the references of today's lecture, so we have taken references from a particular book. The first book is your N.K. Jain of Pharmaceutical Microbiology. Then also your pharmaceutical microbiology Backwell scientific publications. Then some are taken from slide shares, and some are taken from like biotechnology and medicines, bacteria in biology. So you can definitely check out these references for further knowledge. And this is all about today's lecture. Thank you for kindly listening to me.